Hi everyone, greetings. Welcome to our microbiology lecture series. So, by the by, this is Dr. R. Shyam Kumar, Professor, Department of Biotechnology, Kamaraj College of Engineering Technology, Virudhunagar. So, let us start our discussion regarding the microbiology lecture series. The topic is preservation of food. It is one of the topic in microbiology. And the content what we are going to see is principles of food preservation, methods of food preservation, asepsis, removal of microorganism, maintenance of anaerobic condition, use of high temperature, use of low temperature, drying and smoking and other preservative methods. So, coming to the principles of preservation. So, before that, why do we preserve the food materials? Actually, each and every kind of food material which is originated from plants as well as animal sources. That is why there are biological mode which comprises lot of enzymes, at the same time at the surface are normal inhabitant microorganisms also be there. So, that can also oxidize, that can also utilize the substrate present in the food, thereby the food will be contaminated by microorganism or grown by microorganism or by self destruction. So, due to this action the food will be deteriorated. So, it is very much essential to preserve the food material for long time for the consumption of human beings, and also it prevents the microbial infections also. The next thing is the principle. Coming to the principles, the first part is that prevention or delay of microbial decomposition. So, we can suppose to prevent or delay the microbial growth over the food, only then we can preserve the food material. So, that can be done by keeping out of microorganism or by removing microorganism or by hindering the growth of microbial activity. So, these three major action which prevent the growth of microorganism. So, you can preserve the food material and by killing the microorganism. So, we can suppose to apply any process to kill the microorganism. So, further there is no growth of microorganism over the food. This is about the microbial perspectives. And prevention or delay of self decomposition. I already told you that most kind of the foods are derived from plant or animal source. So, they have their own enzymes and biochemical reactions also. So, due to oxidation or reduction potential, what happens, every nutrient present in the food will be exhausted, whereby it will be deteriorated. So, the self decomposition should be prevented by destruction or inactivation of food enzyme. So, native enzymes will be destroyed or the enzyme action will be stopped, or by prevention or delay of pure chemical reaction. That means, the biochemical reactions carried out in the food material will be prevented, whereby we can extend the shelf life of the food material. So, example, the prevention of oxidation process. Usually the biochemical reaction, what happens, oxidation or reduction will happens. So, the same will be stopped or prevented. And next principle behind this is prevention or damage of damage by because of insects, animals or the mechanical causes. So, these three another part it should be prevented. These are all some of the methods which is used to preserve the food materials. And coming to the first one, asepsis. You know what is asepsis? There are two things natural protection as well as artificial protection or manual protection. So, actually natural protection can be happens. So, due to the skin present over the surface of the animal or plant tissues. So, that which prevents the entry of microorganisms or delay or prevent the microbial decomposition, because of the thick shell or thick membrane or thick coating present over the surface of the food material, which can prevent the entry of microorganism. And next is packaging of the food materials. So, we can wrap the food materials without entering of microorganisms into the food material, and application of sanitary methods that which during process either and food industries or dairy industries, during handling there are more chances to have different kind of microbial population which enters into the food, which will contaminate. So, sanitary methods also be followed to prevent the entry of microorganisms into the food material. So, in food industries they are focusing more on microorganism and sanitation process to prevent the entry of microorganism. And coming to the next method, removal of microorganisms. So, in that there are various methods followed to remove the microorganism, that is depends on the type of food materials, either it may be liquid or juices or solid food materials. Different application process may be carried out to remove the microorganisms. So, the first thing is filtration. So, this is one of the successful method for complete removal of microorganisms with the pre-sterilized, see it is important, pre-sterilized filters. So, example fruit juices, soft drinks and water can be filtered, and we can prevent the microbial entry or microbial contamination. And next is centrifugation, usual centrifugation process, industry based centrifugation process may be carried out, but usually in industries this centrifugation process is not widely used. And next is washing, it is another process. The fruits, vegetables and other kind of food materials can be washed with sterile or antibiotic or chemical coated water, whereby we can prevent or kill the microorganisms. And next is trimming. So, we can trim the contaminated or spoiled portion of the food, say for example cabbage. In cabbage at the surface we can trim the skin. So, it can prevent the entry of microorganism. So, this is removal of microorganism. And next method is maintenance of anaerobic condition. So, by maintaining anaerobic condition we can control the growth of microorganism, not only control the growth of microorganism, we can supposed to prevent the self destruction also, that means slowing down of the enzyme action of the food materials also. So, we can maintain the anaerobic condition due to provision of carbon dioxide or nitrogen during after evacuation of the air in the container. So, most of the microorganisms are aerobic and they require oxygen for their growth. So, when you are evacuating the container in the food, which will eliminate oxygen from the container, and while filling with the carbon dioxide and or nitrogen, which prevents the growth of microorganism. So, maintenance of anaerobic condition, which is one of the important method in canning industries or food industries to maintain or to eliminate or to control the microbial contamination. And here the spores are highly resistant to the heat and they can survive in canned. So, even though if we are maintaining anaerobic condition, the spores can withstand. And next is use of high temperature, and here temperature and the time which is used during the process is depending on the effect of heat over the food on the food. Other preservative methods, see after application of the temperature, what preservative method we are going to use, that is another major factor, and the classification based on that we can apply the high temperature to control the microorganism. So, here there are three major processes. First one is pasteurization and heating at 100 degrees Celsius and heating above 100 degrees Celsius. So, this process is being used to control the microorganisms. So, first one we can see the pasteurization. It is one of the major process mainly in dairy industries and some kind of food industries to control the microorganism only vegetative forms. Here it applies 63 degrees Celsius for 30 minutes, especially it is one of the pasteurization process applied in food milk industries or dairy industries, and application of 72 degrees Celsius for 15 minutes, and for juice industries where they can apply 77 degrees Celsius for 30 minutes, and some of the food industries they can also apply 88 degrees Celsius for 30 seconds. So, these pasteurized products, after pasteurization process the cooling process should be prompt, only then it will be preserved perfectly, unless otherwise the contamination will happens or else the self destruction will also be happens. So, the heat process which will not affect or harm the quality of the product. Say for example, if any chemical reaction happens, that may supposed to convert it as a toxin. So, when you are applying the heat, it should not affect the quality of the food, and main spoilage microorganisms are not very heat resistant. At that time we can supposed to go for this pasteurization process. So, say for example, yeast, so they can easily control by pasteurization process. Then to kill pathogens and any surviving microorganism will be treated with other preservative methods. See, due to application of low temperature or during pasteurization process, only the vegetative form of microorganisms will be killed, and other resistant kind of organism can be controlled by some other preservative methods. Then the competing organisms also be killed. The only desired microorganism alone will be facilitated for the fermentation process in industries. So, the pasteurization process is one of the important method to control the microorganisms in heat labile products and fermentation industries also. Then the preservation methods used to supplement pasteurization. Say after pasteurization process, what are the specific preservation technique applied over the food, that is refrigeration. So, we can keep the food under low temperature, keeping out of microorganisms, that means asepsis process, the proper packaging should be maintained, and maintenance of anaerobic condition. This is another major implication that can controls, and addition of high concentration of sugars. So, when you add a high concentration of sugar, it may supposed to prevent the growth of microorganism, then presence of chemical preservatives. So, chemical preservatives also applied in some industries that can controls the microbial growth. And next is heating about 100 degrees Celsius. There are some of the food industries, they can apply a boiling temperature 100 degrees Celsius. There are some methods, boiling, immersion, baking, simmering, roasting, frying, blanching, exposure to flowing steam. So, when you are exposing to the flowing steam, immediate change in the temperature, the vegetative form of microorganism present in the food will be killed. So, this is heating about 100 degree Celsius. And next is heating above 100 degree Celsius. This is obtained by means of application of steam or steam under process, which will makes to eliminate or to kill the microorganisms in the food material. So, in commercially in food industries, they can go for ultra heat sterilization process to control the microorganisms. All commercially sterile food should be stored in cool after sterilization process, and it should be kept in dry place, cool dry place, to prevent any viable thermophilic spores from germinating and to cause spoilage of the food. So, the spore will not be germinated after sterilization process. So, it should be kept in cool dry place. Okay. Then ultra heat treatment, especially in dairy industries, that is about 150 degrees Celsius steam injection, that means flash evaporation. So, that can happen in dairy industries particularly, that will control the microorganisms. And the major objective of the heat treatment in food is to destroy the pathogenic microorganism or spoilage causing microorganism. And next is to destroy the toxin. So, the toxin material which may be produced by the microorganism, that may be deactivated in the food, and to destroy the vegetative cells or spores of the yeast, that may be controlled or destroyed, and to destroy undesirable enzymes. Undesirable enzymes may be secreted by the microorganisms or during biochemical reactions of the food, that will also be destroyed, and the control the growth of surviving microorganism. Already the microorganism may be present at the surface or natural inhabitant of the food material, that will also be controlled when you are applying heating over the food, and to retain the acceptance and nutritional quality of the food. It is very much essential. So, when we apply high heat, it should not destroy the food material while eliminating or killing the microorganism. So, this is important parameter should be maintained or should be focused. So, this is the major objective of applying heat over the food to control the microorganisms. And use of low temperature. Here the low temperature which reduces the activity of microorganism. Actually each kind of microorganisms are very active during optimal temperature. When you are reducing the suboptimal or below optimal temperature, the activity of the microorganisms will be lowered, that means the biochemical reaction will be reduced. So, we can keep the food material at low temperature, which prevents the growth of microorganism. So, microbial growth will be reduced. So, the spoilage will be reduced due to suboptimal temperature. So, when we keep the organism at the optimal temperature alone can grow very fast. So, when you are reducing the temperature, automatically the biochemical reaction of the microorganisms will be reduced. So, we are keeping the food material in the low temperature. It is one of the preservative method, and it will not control the Clostridium botulinum. It is one of the anaerobic microorganism which causes which secretes toxins. And next is freezing temperature. It is another process involved to control the microorganism or to prevent the growth of microorganism. So, when you reduction of microorganisms will not happens due to freezing temperature, but it is not coming under the sterilization process. It can prevent the growth of microorganism, that is at the freezing temperature. And next is next method is drying and smoking. So, drying another method involved to control the microbial growth, because when you are in applying high temperature to dry the food material, that means elimination of moisture content in the food will reduces the water activity of the food material. So, water activity when it reduces, automatically the biological or biochemical reactions will also be decreased. So, the microbial growth will be automatically suppressed or lowered down. Say for example, for Streptomyces rauxi it expects 0.65 water activity. At the same time, Aspergillus glucose it requires 0.6 water activity. So, depends on the organism, the water activity will be varied for their growth. So, when you are applying drying process, it reduces the water activity, ultimately the organism's growth will be reduced. And next is smoking, application of heat with any specific material, chemicals or wooden preservative material. While application of heating, what happens, the water content of the food will be reduced. At the same time, the fumes or smoke will produced from the wood material, which can adheres over the surface of the food material. While what happens, that chemical compounds which is released during smoking process, say for example, formaldehyde or any kind of aromatic or flavonoid, some kind of material which can act over the surface acting microorganism, surface present microorganism. So, that will be controlled, and due to lack of water activity, the microbial growth will be reduced. At the same time, smoking process also enhances the taste and tenderness of the food, say for example, smoked fish. So, this is another process to control the microbial growth. So, these are all some of the methods which is used to preserve the food materials. Thank you very much for watching our lecture series.